Ladies and gentlemen, first I want to thank the organizers and also the Society, of course, for giving me the opportunity to present for you a new model for weight regain after weight loss, the so-called yo-yo effect. These two postcards from the 1960s show you that some decades ago, overweight and obesity was not really recognized as a severe problem because it concerned only a few percent of the population. But nowadays, for instance, in the Netherlands, about 35% of the population is overweight or obese, and this poses a real threat on the healthcare system. Luckily, overweight is not really a problem. It has an easy solution, and that is to lose weight. If you lose already 10% of your body weight, then there is a measurable and significant decrease in the risk for type 2 diabetes and cardiovascular complications. So the only thing you have to do if you are overweight is lose weight. But still there seems to be a problem, and that comes afterwards. And that was nicely expressed, for instance, by the Dutch Obesity Society. They say overweight people really know how to lose weight. They even win prizes for that. What they cannot is to maintain at the reduced weight. So ideally, if you are overweight and you want to lose weight, you follow this green line here. You reduce your weight and you maintain it at this reduced weight for the coming years. But different studies have shown that between 35 and 80% of the subjects, they lose weight, but 1 year after this weight loss period, they are back at their original weight or even beyond. And then the whole process starts all over again. Now, several factors, influences have been identified, and I name the most important ones. That's psychological factors, psychosocial factors like stress or the lack of motivation or persistence to maintain an adjusted lifestyle. There are behavioral influences like repeated snacking or lack of sufficient physical activity to maintain the reduced weight. Some genes have been shown to have alleles, polymorphisms, that seem to predispose for this weight regain after weight loss. And there are also some physiological parameters reported that may be of influence. And one of those is, for instance, the leptin concentration in the plasma. If you lose weight, you lose fat mass, and your plasma concentration will decrease. But it's been observed often now that the leptin, the plasma concentration of leptin decreases more than the fat mass. Now, this effect is thought to cause a state of leptin deficiency in the brain, and that might also contribute to weight regain after weight loss. Now, this observation of leptin, which is produced by the adipocytes, made us think that maybe also the adipocytes play a role in this yo-yo effect. So the question that we asked ourselves is, what is happening at the molecular level in the adipocytes in our body when we are losing weight? And we decided to look into that and look at the level of the proteins of the adipocytes, so we used proteomics techniques. Now, in order to do that, you need, of course, the material. So we took from our subjects needle biopsies from the subcutaneous abdominal fat, just by sticking a needle into the fat tissue and sucking a little bit tissue out. Now, from this tissue then, the complete proteins can be isolated, but of course we want to look at each protein separately. So we decided to use the technique of 2D gel electrophoresis to separate the proteins, first according to their charge, and then next again according to their mass. And what we end up with is a proteome pattern like this, and on average we can see, with the help of the computer, about 1,000 different spots, which means about 1,000 different proteins. Now, spots, as I say, means proteins, but in proteomics we are looking at spots, so if you're interested in a certain spot, you must identify the protein which is present in that particular spot. So, in fact, you should identify all of the proteins in these spots, and that is done by applying mass spectrometry on each of the spots. But I will not go into technical details further here. So we use this technique for our experiment, which is a weight loss experiment, and of course we are comparing two situations then, that is before weight loss and after weight loss. And so we have to make a proteome from the adipose tissue and then compare these patterns and look for the spots, the proteins, which change in abundance due to this intervention study. And the proteins that change, of course, we hope that gives us a clue about the function and about the processes that are changing in the adipocytes. Now, there is one other problem, and Professor Languin already mentioned that also when he was talking about microarrays of the adipose tissue yesterday. That is, if you take a needle biopsy, then you have a mixture of cells. It's not only adipocytes, but it's also epithelial cells. There is contamination of the blood, so there are blood cells, there are nerve cells, and so forth. And we, of course, are mainly interested in looking at the proteins of the adipocytes and not of the other cells. So we try to solve this problem by designing a specific strategy that is a subtraction strategy of different proteome patterns. Now, we do the following. The adipose tissue is, from the adipose tissue biopsy, we make a proteome pattern, but we have also made a proteome pattern of total blood cells and also from purified adipocytes. Now, what we do is that in the pattern of the adipose tissue, we ignore the spots which overlap with spots of the blood cells, and we look only at spots which also occur in the proteome pattern of the purified adipocytes. So this reduces the number of proteins that we analyze in the study, but it gives us a better picture of proteins which are present in the adipocyte, and we call this the adipocyte-enriched proteome. Now, with this technique, we have done the study, so let's now move to the intervention study. This is shown here. We had 8 persons with overweight or with obesity, and so in the beginning of the study, we took the adipose tissue biopsies, and then they were put on a very low-calorie diet for 5 weeks. And during that period, they lost approximately 10 kilos of their body weight. Now, we did not take the second biopsy immediately after those 5 weeks, because then the subjects are still in a negative energy balance, and what you measure then is probably stress in the tissue. So we adjusted their diet to a weight maintenance diet, which was continued for an additional 2 weeks. And after 8 weeks, we took the second biopsy. So we were already very interested to see whether in the adipocytes, in the proteome of the adipocytes of these people who were at a stable dietary phase, if we still could see changes. Now, let's look first at some of the parameters of adiposity. You see here body weight was significantly decreased by 10 kilos, as I said. 7 kilos of that was fat mass. Fat-free mass was also significantly down, so BMI was decreased, and also the waist and hip circumferences. Parameters of the metabolism of these persons, glucose, glucagon, cholesterol, and LDL cholesterol, and the free fatty acids in the plasma all were down significantly. And as a positive control, also the leptin, as you see here, was down by more than 50%. While the fat mass was down by 20%. So this shows already that leptin has a very dramatic decrease with respect to the fat mass. This at least showed us that all the parameters behaved as we would expect from a weight loss intervention. So we were very confident that we did a good job, and we were curious to look at the proteome of the adipocytes. We could analyze about 100 spots of the adipocyte-enriched proteome, of which 40 were identified with the mass spectrometry. Of those 40, 6 changed in abundance and 8 showed a trend for change. Now here are the names with the fold changes of these proteins, but of course it's more interesting to talk about the function than about their names. But 100 spots is not very much, so in the meantime we also have added data by using Western blot, so we added data on targeted proteins, and now we have information about several processes of fatty acid handling in the adipose tissue, in the adipocytes. And you see these processes here. It's about fat storage, fat transportation and handling, it's glucose uptake, and also the beta oxidation, which is distinguished in the mitochondrial and the peroxisomal beta oxidation. You see the proteins which are up and down regulated, and these lines indicate positive correlations, which also shows that these processes are really linked to each other. So we have a lot of information of what is going on at the functional level in these adipocytes. But I'm not going to mention everything. What I want to mention is that we also looked for biomarkers of this intervention process among our proteins. And our definition of biomarkers was that a protein had to change significantly during the intervention study, but also had to change similarly in all the examined persons. So when a protein went up in one person, it had also to go up in all the other persons, and so forth. Now, we were indeed able to find such biomarkers. For instance, 2 isoforms of aldose C. You can see that they all go down in all of the examined subjects. Tubulin, a form of tubulin, goes the other way. It goes up in all the examined persons. And there is also the acidic form, the phosphorylated form of annexin that goes up. But we have only data on 5 of the 8 persons for that. Now, the interesting thing about these proteins which so consistently change in the adipocytes when you lose weight is that if you look into the literature, you can find a process which links these 3-4 proteins together. Now here is a very schematic drawing of an adipocyte with a relatively small lipid droplet, and the part of the cytoskeleton, the vimentin skeleton surrounding it. We have the mitochondrion, the nucleus, and here is also a machinery which is indeed the interesting part. That is the machinery where the GLUT4 vesicles, so the vesicles containing the glucose transporter, are scavenged, are tied to the cytoskeleton. And the link here is made by aldose C, which is in fact mostly known as an enzyme of the glycolysis. But it's also here an anchor for these vesicles, and it ties the vesicles to the actin filaments, and this process is influenced by tubulin. So what we see is upregulation of tubulin, which inhibits the scavenging. We see a downregulation of aldose C, which promotes the release of these particles. And in the mouse, it has been shown that annexin A2, especially the acidic form, is the mediator to translocate these vesicles to the plasma membrane. So apparently what's happening is that the adipocytes are preparing their machinery for the uptake of glucose if you are losing weight. Now this is a consistent process, so this may be the main driver of everything, but we also saw several other things. So the uptake of glucose is apparently promoted, and what we also saw is by pyrrolipin B and fatty acid binding protein that the cells also increase their capacity to recruit fatty acids at their plasma membrane. So they also apparently want to take up fatty acids. Now, we are dealing with adipocytes, so glucose and fatty acids, of course, makes triglycerides, and triglycerides is the form of the fat in which the fat is stored in the lipid droplets. This is not the only thing. I said we see several processes going on. One is the thing that I want to mention here, just to remind me to tell you that the adipocytes, although they seem to go back to uptake and storage of triglycerides, they are not completely returned to their original functional state, because they do not use much glucose for generating their own energy, and that is what they normally do. They are still burning also fatty acids to generate their own energy, which means that they are still in a state of starvation. But they prepare their machineries for the uptake and storage of the triglycerides. So what does it mean? It means that when you are losing weight, your adipocytes are preparing themselves for uptake and storage of fat again. And the question is then, why should they do that? Because there is no lack of energy or anything anymore. And we think that the reason is that they feel cellular stress. And one argument to think that it's stress comes from the further analysis of these proteins that we have found by proteomics. The red proteins are a part of the so-called minimal stress proteome. And the minimal stress proteome is a set of a few hundred proteins that respond to whatever stress you apply to whatever type of cells. So this indicates for us that these cells are in a state of stress, and that stress may be the driving force for the whole process. Now we can start a little bit to speculate where this stress should come from, that they are still feeling in this stable phase. And for that, I want to go to this picture here. It's a picture of an adipocyte. It's with, if you can see it, a lot of dark-colored fat droplets in it. But the important thing is also this stained layer that surrounds the cell, which is called a basal lamina. And so fat cells, when they develop from pre-adipocytes into adipocytes and they store massive amounts of fat, they become very vulnerable for mechanical disruption. And that is because, these cells are a sort of pudding, they're a sort of jelly, and every mechanical stress may rupture them. Now, what they do is, if they develop into a mature adipocyte, they immediately start to synthesize this outer skeleton, this basal lamina, which is a very thick layer and protects the cell from this mechanical destruction. Now, this this extracellular matrix, the synthesis or the modification, we found out that it's a very energy-costing process. For instance, the synthesis and modification of all the collagens in this layer, they cost a lot of ATP. And this is just shown by our analysis here. Here we show one specific process. It's the modification of the collagens at the prolines, so the proline hydroxylation, which is necessary for the collagens to allow them to multimerize into these fibrils. Now, what this process needs is the cofactor alpha-glutarate, which is then converted to succinate. But this means that if you look at the TCA cycle, that alpha-glutarate that this process makes a sort of shunt from alpha-ketoglutarate to succinate, and it skips the possibility to make 4 ATPs in the TCA cycle. So in other words, one proline hydroxylation costs already 4 ATPs. And there are a lot of prolines in a lot of collagens to make this extracellular matrix. So it costs a lot of energy to maintain this and to modify it. Now, this made us propose then a model a cellular model for the yo-yo effect, because what we feel is the following. We feel that the adipocyte, if you start to lose weight, it gets rid of its fat, of its triglycerides, which means that the volume of the cell will shrink. And that requires that the surrounding extracellular matrix also is modified accordingly. But in a state of starvation, if you do low caloric intake, then this is not possible. So mechanical stress will build up between the cell and the surrounding matrix. And the most easy way for the adipocyte to get rid of this stress, to overcome this stress, is to go back to its original volume, and that is by storing fat again. Now, we even think that it might be so that since adipocytes send out a lot of adipokines, one of them is leptin, to regulate in the periphery and so forth, and in the brain, the energy uptake and energy usage, we also think that this stress may change the adipokine pattern of the adipocytes to induce in the carrier of these stressed adipocytes increased energy intake, so that the adipocytes can go back to their relaxed state. Okay, this is the model that I wanted to present for you. We still have, of course, to do a lot of validation and to prove whether the different parts of it are actually true. But I think we have added another dimension to the possible causes of this yo-yo effect, and that's the adipocyte stress. So finally, I want to thank the people from Maastricht University who contributed till now to this work, but also want to mention Quinn Eastman, who wrote a very nice editorial about our work in the Journal of the Proteome Research, and this made from that editorial now people call this model the model of the screaming adipocytes, because we said that the adipocytes are stressed and they are screaming for fat again. Okay, thank you very much.